a unique school based on Indian cultural heritage and global vision, which has a record of 100% result in ICSE, State Board and PU Board examinations. Athreya Educational Institutions, a famous name in Mandya for more than a decade, providing you a world-class education from nursery to PUC. Enhanced with a newly constructed environment-friendly building, spacious smart classrooms, well-equipped labs and library, experienced teachers with motherly care and updated teaching methods, including 4-language learning concept, regular multiple cultural and sports activities, and best vehicle facility. What else do you expect? Join your children today. We make them complete human beings and present them as real gift to the society.
ಶ್ರೀಮತಿ ಅನುರಾಧ ರಘು ಅಧ್ಯಕ್ಷರು, ಕೆಎಸ್ ರಘು ವಕೀಲರು ಹಾಗೂ ಕಾರ್ಯದರ್ಶಿಗಳು. ಅಡ್ಮಿಷನ್ಸ್ ಓಪನ್ ಫಾರ್ 2020 and 21. Pre-KG, UKG classes to 10th standard. ಸಂಪರ್ಕಿಸಿ ದೂರವಾಣಿ ಸಂಖ್ಯೆ 9986797652, 8867015936 ಹಾಗೂ 8867015937.
Experience Athreya. Feel at home.
Good day to all of you. So since we have met in the previous session that we discussed about the structure and the function of cell and its basic, important organelles which is present inside them, and what is their role in the cell, and what kind of a structure they have, and all those stuffs. So now today's class we will talk ahead continuation of that same content, and we are going to talk about the chromosome. Before we get into the topic chromosome, we understand where actually it is situated and what is its structure and what is its important function. Before that, let us get into this important part of the cell, that is a nucleus. When we spoke about the three important structure of the cell, we spoke about the plasma membrane, we also got introduced to the cytoplasm, and also we got introduced to the important structure, that is a nucleus. Nucleus is a very key important structure which plays a vital role in controlling and regulating the important organelles present inside the cell. Now we know, like inside the cell there are so many different organelles present. They have their structure, they have their function, and this entire structure which is present inside the cell, that is called as organelles. Okay, these organelles is regulated. The work of the organelles is being regulated by the nucleus. How does this nucleus look? Nucleus is a round, dense structure which is present in the mid of the cell. In the animal cell, usually you find the structure of the nucleus in the exactly center position, but in the plant cell it is slightly pushed towards the wall. Now let us understand the structure of the nucleus. So the structure of the nucleus carries important parts. That is the outermost part, which is called as a nuclear membrane, and nuclear membrane are double membrane. Okay, so the nuclear membrane are actually doubled membrane. They are mainly helps in the shape of the nucleus. It helps in the framework of the nucleus. Now inside the nucleus it is a nucleoplasm. Now let me give you the clarity. This is nuclear membrane. Okay, in the nuclear membrane you can actually see there are small tiny pores which involves or which helps in the movement of certain important particles in and around the cytoplasm. So there are certain particles which can get into the nucleus, there are certain particles which can move out of the nucleus, and that important tiny structure is called as a nuclear pore. Pore is a small tiny hole like structure which is present around the nuclear membrane. Now, apart from this, there are some more important structures which is present. There is a dark dense structure which is present inside the nucleus towards a corner. There can be one or two in their number, which is called as a nucleolus. And we have a content, the semi-liquid content, which is filled around the nucleus, just like a cytoplasm around the cell. There is a liquid content which also present inside the nucleus, which actually gives her the proper shape for the nucleus, and that structure is called as a nucleoplasm. Okay. Apart from these important structures, there are very minute, a thin structure which is present, and they are nothing but chromatin fibers, which is a thread-like structure. Now, this is what the most concerned part of the nucleus and the most important part of the nucleus, and this is a very thin thread-like structure called as a chromatin fiber. Okay, so today we started with introducing to the nucleus, because nucleus plays a very important vital role in the cell. It controls all the important organelles present in the cell. It coordinates the cell to perform important function. Okay, and a nucleus is a round dense structure which is present in the center portion of the animal cell and towards slightly corner into the plant cell. When we look to the depth of the structure of the nucleus is made up of double membrane structure which covers the entire nucleus, which is called as nuclear membrane. On the nuclear membrane, there are small tiny holes studded around, and these small tiny holes helps in the in and out movement of certain materials, which takes few informations or ion exchanges or few mRNA production, RNA, protein synthesis, proteins, enzymes. There are so many substances which move in and out of the nucleus, and this is achieved with a small tiny hole which is called as nuclear pore. Apart from this nuclear pore, there is a dense semi-liquid content which fill the nucleus. That is why the nucleus is very fluffy in its size, and that is just because of the nucleoplasm. Nucleoplasm and cytoplasm are a same kind of a composition, but the liquid present in the nucleus is said to be the nucleoplasm. If it is out of the nucleus, is said to be the cytoplasm. Then, apart from these structures, we have a dense dark stained portion which is called as a nucleolus, which is present in the nucleus, and this nucleolus is mainly responsible for the production or the synthesis of ribosome. We will be talking along like what is these important function of these ribosome. I think we studied even in the previous class also. Ribosomes are mainly responsible for protein synthesis. If a protein has to get synthesized, ribosome plays a very important role. Without ribosome, the protein synthesis will not, you cannot achieve synthesis of protein. So that ribosomes is actually produced inside the nucleus. Ribosomes. This helps in and out movement. Of particles. Nuclear membrane is protective in function. Okay. Nucleoplasm is a semi-liquid which helps in diffusing of few of the materials into the specific portion of the nucleus and also exiting out of the nucleus. The most important part of the nucleus is the chromatin fiber. Now we are talking about a chromatin fiber. Chromatin fiber is a long thread-like structure. It's a thread-like structure which occupies inside the nucleoplasm, and they are just scattering without any a restricted position. They are just scattered around the nucleoplasm of the nucleus. Chromatin fibers, chroma means, please understand, it is nothing but the substance which takes up the color. That is why they are called as a chromatin fiber. These fiber takes up the color when we introduce them to staining process. So that is why they are called as a chromatin. And nucleus has this important structure, chromatin fiber, where the hereditary material are present inside these chromatin fibers. Now we are going to highlight this important structure, that is chromatin fiber. So we will get into that discussion now. Hope you understood about the nucleus. Nucleus plays a very important role and its important structure, and their function is discussed. Now we will talk about chromosome. Now what do you mean by chromosome? We spoke about chromatin. What is chromosome, where it is present, and what is the connection between a chromatin fiber and the chromosome? Now, chromatin fiber. I told you chromatin fiber is a long thread-like structure. It is a long thread-like structure. Okay, it is present around the nucleoplasm of the nucleus. This long structure is so long. Okay, these long structures called as a chromatin fiber, and they are present in the cell whenever the division is not occurred. Okay, when the cell is getting into the step of a division, that means if a cell is attaining a division process, these chromatin fiber, now this is a chromatin fiber, let us say like this is a chromatin fiber. This is a long structure which is present inside the nucleus. Okay, and these chromatin fiber undergo condensation. Condensation means they get coiled and supercoiled and form an important structure which is called as a chromosome. Now I am just giving you a very rough idea. This is not the structure of a chromosome, but later on these coiling, so they undergo coiling. Okay, this is chromatin. These are chromatin fibers. They undergo condensation, and this condensation, they are condensed. That means it is achieved by coiling process. That means a long thread-like structure undergoes coiling. Again, these structure undergoes supercoiling. Okay. Hope you are able to understand. You see, just like a telephone cord, you have seen a telephone wire, isn't it? Now we have these mobiles. There are few places where you can actually see telephone wires, and they have this kind of coiling structure. It is again coiled, double coiling. Okay, so it is coiled now, double coiled. They are called as a double coiling. Again, there are a few more important, further coiling takes place where, okay, like this, it is again called as a supercoiling. Just like a solenoid, you can see the structures which are supercoiled. This is called as a supercoiling. Okay. So this long thread-like structure, they are 2 meters long. Please understand, they are 2 meters long, and this long thread-like structure is accommodated inside the small tiny cell, that too inside the nucleus of the cell. Okay, the cell itself is invisible and the structure of the nucleus is invisible, and inside the structure of the nucleus, these long thread-like structure is present, and they are present in a condensed form. And that is why these long structure undergoes condensation, that is through coiling process. Again, this coiling is not sufficient. They undergo double coiling and supercoiling, which result in a fine structure which is called as chromosome. I have just written in a very rough fashion. This is how the final structure is achieved. So this is how the coiled portions gets into, and they are organized in such a way that they look a final, finally they actually look in this fashion. So this is how the structure of chromosomes are represented. Okay. Now this is called as a chromosome. Now, did you get the connectivity to the chromosome? Chromosome is nothing but a condensed structure of chromatin fiber. Chromatin fibers are present inside the nucleus of a cell, and they undergo condensation only when the cell attains cell division. And this structure is achieved only when the cell attains cell division. If the cell is not undergoing cell division, then the structure is chromatin fiber only. When the cell is getting ready for the cell division, these chromatin fiber undergo condensation to finally form chromosome structure. Okay, hope you understood till here. Now let us understand what is actually present inside the chromosomes. Okay, chromatin fiber and chromosomes are nothing but same, but chromosome is a well defined structure where there are supercoiling structures of this chromatin fiber. Now please look into this diagrammatic representation. Now I am going to tell you what are the structures which actually present inside the chromosome. Chromosome, I told you how this a structure is formed. That is just because of the repeated condensation, coiling and supercoiling structure of the chromatin fiber. How this coiling takes place? Who is responsible for this coiling? And what actually is present in the chromosome? We can just read it out now. Now let us understand that. Okay, a chromosome structure is enlarged here. If you see the chromosome structure, there are two important structures present. It's an arm. This is called as the arm of the chromosome, which is called as a chromatids. There is upper and lower arm. Okay, these two arms are connected at the center, which is called as centromere. Okay, chromosome has two sister chromatids, upper and the lower, and these two chromatids are connected at a junction which is called as a centromere. So it is centrally, it is holded in the center position which joins two chromatids, that is called as a centromere. Okay, if we enlarge this, okay, this one portion bigger, we see the coiled portion. This is a supercoiling portion of the chromatin fiber. If we enlarge that into a bigger, you see there is a coiling structure, and this coiling structure again undergoes solenoid kind of arrangement. Okay, like this, you see, there is a systematic arrangement of these chromatin fiber in a very fine structure. And this is called as a chromatin fiber. They are just packed inside the chromosome. Okay, these chromatin fiber, if we keenly observe what actually present in this chromatin fiber have two important components. Okay, chromatin fiber is made up of two important components. The one is DNA. What is DNA? DNA is deoxyribose nucleic acid. Okay, It is the chromatin fibers, that is this is present inside the chromosome, because chromosomes are the content structure of the chromatin fiber. This chromatin fiber have two important components. The one is DNA. It is nothing but deoxyribonucleic acid, and the amount of DNA which is present in the chromatin fiber is just 40%. The total chromatin fiber is covered with 40% of the DNA. Okay, this DNA undergoes winding, coiling. All this coiling is done by the DNA with the help of another important component, which is called as a protein. What is the name of the protein? And this protein makes up 60% in the entire chromatin fiber. So more than DNA, it is a protein which is present inside the chromosome, and these protein are called as a scaffold protein. Scaffolding means they helps in coiling of the structure, and this scaffold protein has an important name, and that name is called as histones. Hope you understood. Chromatin fiber, this entire chromatin fiber has made up of two, it has two important components. One is a DNA, that is deoxyribonucleic acid, and the content of the DNA present in the chromatin fiber is just 40%. Apart from this, we have a special protein which helps in scaffolding of this DNA. Scaffolding means, coiling process. It helps in the coiling of the DNA or winding of the DNA. Coiling of the DNA is actually done by a scaffold protein, and the name of the scaffold protein is a histone, and the total amount of scaffold protein in the chromatin fiber is 60%. Okay, hope you understood this part. This is very important. Now if you look here, DNA and histone collectively called as nucleosome. So this is an important structure. They might ask you, define a nucleosome. Nucleosomes are nothing but the combination of both DNA and histones. They are collectively called as nucleosome. So we'll wrap this portion now. We will talk about structures. So we have DNA and we have protein in the chromatin fiber. Okay, these both are collectively called as nucleosome. Understood? So this is about the structure. Now if you ask who discovered DNA, DNA was first discovered by Walter it is Rosalind Franklin. Okay, Rosalind Franklin actually discovered the DNA for the first time inside the cell, but the complete structure of the DNA was given by Watson and Crick. Watson and Crick. At the same year, that is 1953. Okay, in 1953, Rosalind Franklin told that DNA is present inside the cell, and they are the major important hereditary character present inside the cell, and the complete structure of these DNA was exhibited, or it was published or discovered by Watson and Crick, and the structure of the DNA is called as a Watson and Crick model. So the structure of DNA is otherwise called as Watson and Crick model. Okay, and that structure was so innovative, and it gave a complete clarity about the DNA structure, thereafter the gene, thereafter so many different learning and research were achieved due to the structure of a DNA. And for this they were awarded Nobel Prize in the year 1965. In the year 1965 they were awarded a Nobel Prize for Watson and Crick for their complete structure of DNA, and that structure is also called as DNA double helix. We will learn about this structure next. So this is about the complete structure of the chromosome and what are the important components present inside the chromosome. So just a recap, chromosomes are nothing but the condensed structure of the chromatin fiber. Okay, this chromosome structure is achieved only when the cell attains cell division. Till then, if the cell is normally present, they will be in the form of chromatin fiber. But if the cell is about to undergo cell division, then they attain the structure of chromosome. And this chromosome is supercoiled and multiple coiling, which is made up of huge amount of or huge number of nucleosomes. Millions of nucleosomes are present inside the chromosome. What is a nucleosome? When we talk about nucleosome, so this is a nucleosome. Nucleosome is made up of two important components. They are DNA and histone. DNA makes total 40% of the nucleosome, that is chromatin or chromosome. DNA is otherwise called as a deoxyribonucleic acid. And protein helps in scaffolding, that is coiling of the DNA is achieved by the protein. They are called as a scaffold protein. Otherwise the name is called as histone, which makes the 60% of the total chromatin fiber. And the first time the DNA was discovered by Rosalind Franklin in the year 1953, and the structure was given by Watson and Crick. And the structure which was proposed by Watson and Crick is also called as Watson and Crick model of DNA. They were awarded Nobel Prize for their wonderful, remarkable achievement in giving out the complete detailed structure of the DNA, and it is also called as DNA double helix. They were awarded Nobel Prize in the year 1965. I hope you understood the concept about chromosome. Okay, just we understood about the complete structure of the chromosome and also how the structure determines with the DNA, how the connection goes with the DNA. Now, actually the chromosome structure was first proposed or given by Walter Fleming. Okay, these important Fleming. Okay, that should be one clarity. And DNA, the shape of the DNA was given by Rosalind Franklin, and the detailed structure, the complete structure was given by Watson and Crick. Sorry, it is 1962. I think I said 1965. It was in the year 1962. They were awarded a Nobel Prize for their remarkable work on the structure details of the DNA. Now let us understand about DNA. DNA, okay, let me talk about the nucleosome first, and then we will get into the structure of DNA. As we said, nucleosome comprises of two important components, that is DNA plus histones. Okay, DNA is a negatively charged particle while histones are positively charged particle, and the attraction between these two makes the DNA to undergo coiling around the histones. Histones are the specific structure. There are four histones which is present in the form of a dimer. It is a 4 histone. Okay, like this. At the back end we have another 4 histones. Hope you are able to understand. And this is the complete structure of histones. Histones are round in its the protein structure, which is helping in scaffolding DNA, that is helps in coiling the DNA. So this is called as, see, each histone we have 1 2 3 4 here, and the same histones are repeated 1 2 3 4. They are present in dimer. Okay, it is called as histone dimers. All 4 histones will be in 2 numbers. So this histones, this structure helps the DNA to undergo coiling. Now DNA surrounds the histone by making one fine coiling. Okay, this is one coiling of DNA around the histone molecule. So we have 8 histones, hence it is called as histone octamer. 8 histone, that is 8 histones helps coiling of DNA. Okay, 8 histone will become a one nucleosome, a molecule with a DNA wrapped around it. So 1.5. Now if this is a histone, the DNA goes around and one more half. So 1.5 coiling takes place around the histone molecule. So like this, these nucleosomes are present. Millions of nucleosomes are present inside the nucleus, in which the DNA keeps on undergoing coiling. Did you understand? So this is how the coiling and supercoiling takes place, in which the histones helps in scaffolding or winding of a DNA to make the DNA condensed in its structure. So this is about the nucleosome. Now let us understand what is present inside the DNA, like what is the structure of the DNA and what are the components which is present inside the DNA. This is about nucleosome. Now let us understand about DNA. DNA is called as the deoxyribose nucleic acid. Okay, so it is an acidic component which is present inside the nucleus, and this deoxyribose nucleic acid is a macromolecule which is made up of, it is a macromolecule which is made up of repeated nucleotides. Okay, repeated nucleotides. Now what is nucleotide? We will talk about that in short. So we spoke about what is a nucleosome and what the nucleosome comprises of, and how this histone helps the DNA to undergo winding around it and making the long length of the DNA to get condensed. So the nucleosomes are millions, there are millions of copies present, they are in millions. Okay, since they are in millions, the long 2 meters of DNA is coiled and supercoiled, and it is packed inside the small tiny nucleus of the cell. Now what does actually this nucleic acid, that is deoxyribonucleic acid, has? They are the macromolecule structure. Okay, they are made up of so many, they are the long structures which is present inside the cell, and they are made up of repeated nucleotides. Now what do you mean by nucleotides? Now let us understand. Nucleotides means they are the combination of pentose sugar, okay, phosphate group, and nitrogenous base. Okay, The nucleotide are nothing but it's a combination of three important components, that is the pentose sugar. Pentose sugar means it is pentagroup sugar, okay? It is a pentose sugar, and we have a phosphate group. It's a phosphate group, and we have nitrogenous base, and in nitrogenous base it's a 5-carbon ring sugar that is called as a pentose sugar, okay? It is 5-carbon ring sugar. 1 2 3 4 5. Got it? So it's a 5-carbon sugar, and this 5-carbon sugar will be connected with the phosphate group, and this phosphate group is linked with these, and also this pentose sugar is linked with nitrogenous base. Now what do you mean by nitrogenous base? They are the nitrogen component, they are the nitrogenous cyclic components which are present inside the DNA. They make the DNA actually. They are the very important structures which is present inside the DNA. Nitrogenous base are of 4 different types. We will talk about that. There is adenine, guanine, okay, cytosine and thymine. Okay, these are the 4 nitrogenous base present in the DNA. We are only talking about DNA. So these are the 4 nitrogenous base which is present inside the DNA. So DNA is made up of repeated nucleotides. They are the macromolecules, that is, they are the large molecules present inside the cell, and these nucleotides are made up of 3 important components. First one is a pentose sugar. They are made up of 5-carbon ring sugar, and second is they have phosphate group in them, and also third one, they are made up of nitrogenous bases. There are few bases which is made up of nitrogenous substance, and they form the DNA. Now nitrogenous bases are of 4 different types, that is adenine, guanine, cytosine and thymine. Okay, now adenine is represented by A, guanine by G, cytosine by C capital, and thymine by capital T. Okay, so this is what we have we have understood for today's class. These are the structures which is present inside the DNA. So we have adenine, guanine, cytosine and thymine. These are the 4 important bases which makes up one of the larger part of the DNA molecule. I think we will end it for the day. Thank you so much.
A unique school based on Indian cultural heritage and global vision, which has a record of 100% result in ICSE, State Board and PU Board examinations. Athreya Educational Institutions, a famous name in Mandya for more than a decade, providing you a world-class education from nursery to PUC. Enhanced with a newly constructed environment-friendly building, spacious smart classrooms, well-equipped labs and library, experienced teachers with motherly care and updated teaching methods, including 4-language learning concept, regular multiple cultural and sports activities, and best vehicle facility. What else do you expect? Join your children today. We make them complete human beings and present them as real gift to the society.
ಶ್ರೀಮತಿ ಅನುರಾಧ ರಘು ಅಧ್ಯಕ್ಷರು, ಕೆಎಸ್ ರಘು ವಕೀಲರು ಹಾಗೂ ಕಾರ್ಯದರ್ಶಿಗಳು. ಅಡ್ಮಿಷನ್ಸ್ ಓಪನ್ ಫಾರ್ 2020 and 21. Pre-KG, UKG classes to 10th standard. ಸಂಪರ್ಕಿಸಿ ದೂರವಾಣಿ ಸಂಖ್ಯೆ 9986797652, 8867015936 ಹಾಗೂ 8867015937.
Experience Athreya. Feel at home.